Hi, today we will be discussing about plant genetic resources. This is a very important topic in the course Sustainable Management of Biodiversity. This will form a bedrock for further understanding in agrobiodiversity, conservation and management, and this is very important as far as our agriculture development and growth is concerned. I would like to start with Vavilo. He is considered as the father of plant genetic resources. He said the theories of source material and origin of cultivated plants must be fundamental for scientific plant breeding. In other words, what he said was the cultivated plants was originated from a region wherein the close relative of the domesticated plants were present, or in other words, in that particular region there was lot of diversity, and that particular region was called as center of diversity. N.I. Vavilo gave an important concept in plant breeding, that is center for origin, center for domestication, and he said the concept was very simple. He said simply that in certain regions there is diversity, maximum amount of diversity is there, and in that particular region the crops have been domesticated. And if you see, he said two important centers. One is the primary center of origin wherein the maximum level of diversity existed, and from that particular region the crops domesticated, the crops originated, that has been passed on, moved to some other place due to human action, and also the basic purpose of the movement of the crop material or crop is for agriculture purposes, for settled agriculture, and thereafter after moving to a new place the particular crop was grown for time and again for a long number of years, then that particular region, that particular center is called secondary center of origin. In the secondary center of origin the level of diversity is not much, it is not like primary center, the diversity level is less in secondary center of origin. However, the secondary center of origin is very important from the point of view of conservation, from the point of view of plant breeding it is very important, because in this secondary center of origin one can find primitive varieties, one can find land races. We will discuss about land races in some time from now. And as far as plant genetic resources is concerned, the topic which we are discussing now has earned currency for simple reason that the population pressure is increasing, and because of increasing population pressure the need for agriculture production, increased demand for agriculture production, that is fast increasing, and simultaneously if you look at it, there are the kind of the modern agriculture, the kind of stress the modern agriculture faces is tremendous. The stress in the form of biotic stresses, in the form of abiotic stresses, the stress is too much, and there was always urge for increasing the production, urge for increasing the yield. So in that case we need to understand to stabilize the yield under extreme or unfavorable conditions, under adversary conditions, meaning to say that the production, agriculture production has to be increased in an era of climate change, or in an era of extreme stress, or in an era of population explosion. So under these circumstances there is no other way out, we need to go back to plant genetic resources. The meaning to say that the cultivated plants whatever we are having now, it has been originated from plant genetic resources, and these plant genetic resources is the repository of genes of all the beneficial qualities, traits. So the basic need for this plant genetic resources we have understood now, and it is the right time that we need to explore the plant genetic resources and collect the whatever the resources we are having it and conserve it. So that is the reason why this particular topic is important and in the canvas of sustainable management of biodiversity. And straight away if you look at it, the basic the underlying meaning of plant genetic resource, if you look at it, it means that the genetic information encoded by the genomes. It includes the biological mechanisms for translating that genetic information into phenotypes, and the form and the level of biological organization whereby the genes and the phenotypes perpetuate. When I say perpetuate, it means the organs, reproducing organs, seed, and it is all perpetuate, and this is in nutshell plant genetic resources. This is in a nutshell the genetic information, and that genetic information has to be used for the benefit of mankind, for the existence of mankind, for the survival of mankind, and that is the reason why it is called as resources. So, plant genetic resources is a very, very important cornerstone in agriculture management. And when you look at the importance of plant genetic resources, it is a very, very important thing, and if you look at it, the present day cultivated crop plants, there are around 500, almost 500 crop plants have been domesticated, and most of the high yielding varieties, high yielding crops, if you look at it, it is having a bottleneck. When I say bottleneck, it is genetic bottleneck. When I mean it, what I mean to say is that the modern cultivars are having narrow genetic base. Narrow genetic base makes the crop plants vulnerable, genetically vulnerable to stress. It can be pest, stress due to pest, or a pathogen, or climatic stress, or even the soil environment, or even the mineral stresses. So, in nutshell, the plants, the crop plants are vulnerable. Their existence is under stress, I mean, it is difficult. So, one more important point I wanted to stress here is these cultivated crop plants, they whatever the characters, whatever the traits they are having, it is more suitable for the mankind. So, we have gone for domestication, we have selected the crops, whatever the traits suitable for our need, to meet our needs, we have developed this particular cultivated crops. So, in other words, these cultivated crops are susceptible to myriad number of stresses. And if you look at it for an example, the southern corn leaf blight epidemic, it is a very major epidemic that has occurred in United States of America way back in 1970. It has caused such a devastation, completely corn crop was devastated because of this particular fungal organism, that is Helminthosporium maydis. This particular organism caused devastation. Why? Because the cultivated maize crop was vulnerable. It had a narrow genetic base. Why? Because if you look at it, all the hybrid crops grown during that particular period, they had a particular gene, cytoplasmic gene, and that particular cytoplasmic gene was vulnerable to a particular race of Helminthosporium maydis, and this is the reason why the southern corn leaf blight epidemic occurred in US in 1970. And also if you look at it, one more important disease that has occurred in India way back in 1943, that has caused Bengal famine, that is called brown spot disease of rice. The causal organism is Helminthosporium oryzae. It has caused a famine. What else I can say? So, the reason, the prime reason is our cultivated crops had a narrow genetic base, and just because of that thing, they were vulnerable to the stresses. And second important point, as far as the plant genetic resources are concerned, we need to understand the importance of local land races in plant breeding. Local land races, its importance must not be underestimated. Why? For the simple reason that it is a repository of all the beneficial characters. Of course, all the plant genetic resources is a repository of genes, good quality genes, but as far as the local land races are concerned, it is existing and it has been developed by the farmers for so many years, in hundreds of years. They have developed the particular race, land race, they are primitive varieties, and that primitive variety has accumulated all the good traits, and that particular land race can be looked for at times of adversaries. When I say at the time of stress, we can look for that local land races. One important example I would like to stress at this point of time, in the Mexico way back in 1960s and 70s, semi-dwarf varieties, wheat varieties was developed, and that was directly introduced in Mexico. fine, the crop was started growing, but unfortunately that was affected severely by rust diseases. Both stripe rust, that is Puccinia striformis, and Puccinia graminis variety triticea, both rust caused devastation. Why? The variety was very good, it was high yielding semi-dwarf variety, but it caused devastation because the variety, whatever the variety that has been introduced, that was not taken, they have not taken into account the kind of susceptibility, they have not taken into account. See, immediately what they did, the plant breeders in Mexico, they started breeding the high yielding semi-dwarf varieties with the local land races. Once they bred and the progeny, they started cultivating it, and the progeny had resistance. So such is important, such is the importance of local land races. We need to understand the local land races and we need to use it, we need to use it, incorporate in plant breeding activity. That is how we can proceed further for in the advancement of agriculture. And second important example as far as local land races, where I wish to say, that is a pearl millet variety. Pearl millet is an important crop in India, and it was developed, bred in India, and it was introduced in the western part of Africa. There again, the particular pearl millet variety, that was affected by mildew diseases, fungal disease. Mildew means it is a fungal disease, and that particular pearl millet also, it was susceptible to this mildew problem. So what is that they did was, they started taking a local land races which was having the resistance to mildew, and they started breeding the local land races with the high yielding variety of pearl millet, and thereafter they released the variety, and which was found to be resistant to the mildew. So that is how we need to incorporate the best possible resources available in the complete ambit of plant genetic resources. And a very important thing, after the importance, we have come to know that how it has to be, what exactly the plant genetic resources, what role it is going to play in not only in the plant breeding, for the agricultural domain completely. So when you look at it, the PGR, the plant genetic resources, can serve as a keystones for a natural and man-made ecosystem. If the PGR, if you consider it as a VD crop plant, VD plant, and definitely it can act as a keystone of a natural and man-made ecosystem. And second important point, as far as the role of PGR is concerned, the PGR forms the bedrock for human economies and subsistence. The point here is, for human existence, for higher agriculture production, this plant genetic resources must be conserved, must be looked for, and we need to take up the maximum possible the good genes, or the genes, the traits that has to be taken from the PGR. That is the reason why I said it is very, very important for human economies and subsistence. Thus PGR has formed an important component of research infrastructure, because the kind of research, the basic and applied crop science research, for that particular research to proceed, the PGR is very, very important, and at times this PGR forms an experimental subsystem also, and it is forming a subsystem for further research. This PGR is used, and we know fully well that PGR aids in improving the traditional agriculture systems. The traditional agriculture systems, we need to look carefully, for the simple reason that the traditional agriculture system many times are sustainable in nature, and for that reason we need to understand the traditional agriculture systems, and we need to improve still further, and we try to implement it. We try to encourage a kind of a synergy between the traditional agriculture system and the modern agriculture system. So for that reason the PGR must be definitely looked for. Next important point is, the crop PGRs are used in improving crop cultivars. This crop plant genetic resources or crop germplasm are used in improving crop cultivars, that is very natural, and for developing even new crop varieties also, this crop germplasm is used quite often. And coming to the conservation, because I have talked about the importance, I talked about the role of plant genetic resources. We need to understand that how the world over, the people, the scientists, and how what steps we have taken for conserving the plant genetic resources. Way back in 1961, looking at the importance of plant genetic resources and how that can be helpful for feeding the growing population. Food and Agriculture Organization conducted a high level international technical meeting on plant exploration and introduction, and thereafter immediately they established a cell for purpose of plant genetic resources, and that included not only the plants, it included the forest crops as well. So that was the importance given to PGR conservation, and immediately thereafter an important board, that is International Board for Plant Genetic Resources, was established. It is a very important watershed movement or watershed event in PGR conservation, because this particular establishment of IBPGR was instrumental in furthering the conservation activity. Furthering at the global level, it was playing a very important role. So the basic role played by this IBPGR was to monitor at the global level the plant genetic resources activities. And second important activity of IPBGR is promotion, collection, conservation and evaluation of PGR of species of economic importance and their wild relatives. So this is how the PGR conservation, it was, the momentum is in a higher speed, because now world over there are a lot of organizations looking for conserving this PGR. At the nation level also they are conserving the PGR for the future generation and for the posterity as well. And when we are talking deeply about the plant genetic resources, I was talking about the variability. I was talking about the variability that is existing in plant genetic resources. What is the reason? What is the prime factor? What is the driving factor for this variability? Why there is a variability in this plant genetic resources? Remember, only when there is a variability, a crop can be evolved, or in other words, evolution of crop plants is possible only when there is a variability, and we need variation. Only when we have the variation we can go further for selection, or we can go further for any plant breeding activity. So this genetic variability of crop plants is a cornerstone for plant breeding, and this variability is a product of gene mutation, migration, recombination, selection and genetic drift. We know the mutation is a sudden heritable change that is taking place in the organism, and it can be perhaps a macro mutation or a micro mutation, but this mutation is the foremost important event or cause for variability, and just because of this mutation domestication itself was successful, or in other words genetic variability was possible because of gene mutation. And that mutation, when I was talking about the macro mutation, it is the kind of the phenotypic change that will be of larger extent, you can find it out, that is about macro mutation. But as far as micro mutation, it is not tangible, but however the mutation is taking place at the quantitative level. When I say quantitative traits, smaller level, that is micro mutation, but both macro and micro mutation was instrumental in creating variability, and that variability, because of that reason we can go further for selection or developing new crop plants, or we can use that particular resources for plant breeding activity. And second important reason for genetic variability is migration. When I say migration, it is a movement of population or movement of an individual from one population to another. This particular movement, the individual moves from one population to another. After reaching to another population, it starts involving in reproductive activity. So because of this reason, because of this migration process, what happens, there is a change in the allele gene frequency, genotype frequency. Because of the introduction of an allele, there is a change in the gene frequency, and that is causing variability. That is causing variability, and that is very important thing. When you look at the plant breeding, the normal parlance, if you see the intervarietal crosses or polycrosses, that all signifies the migration activity, and this migration is causing the change in the gene frequency. And next important factor or driving force for causing variability is recombination. When I say recombination, that is a product of that segregation that is taking place in the allele level, and because of this kind of segregation and formation of a new product, new genotype, because of that what kind of thing benefit we are getting? We are getting the benefit of variability, a high level of variability we will be getting because of recombination, and that is what taking place at the allele level. Next important thing is selection. Selection is a very important phenomenon that is responsible for causing variability, and when you look at the selection as such, it means what? It means that non-random differential reproduction of genotype is selection. It can be either a natural phenomenon or artificial or driven by human activity. When I say it is a natural selection, if it is a selection is a natural, it means that it is occurring in a natural population and driven by a natural factors like the climate, the environment, or even the soil. So this kind of factors were responsible for selection, the process of selection, that is called natural selection. The byproduct of natural selection, if you look at it, is the variability will be very wide, but on the other hand, if the selection is taken care of by an artificial agent, in that case what happens? The variability is narrowed down. When I say narrowed down, it is having the kind of variability is very less. So our main focus as far as selection is concerned, many types of selection is there, disruptive selection, directional selection, and even stabilizing selection. So these all selection has to be taken into account for understanding the process of creation of variability in croplands. And next important phenomenon, that is a genetic drift or a random drift, and this also causes change in the gene frequency due to a sampling error. It is occurring due to randomly because of the sampling error, and this genetic drift is more often found in case of small population. In the case of small population, the error will be more, the sampling error will be more, and because of this reason what happens, the allele, that frequency, that changes, and as ultimately if you look at it, the allele frequency in a particular allele, it will be 0, and in another case it will be 1. When I say when the allele frequency it is moving towards 1, what happens? There is a fixation that is what taking place in genetic drift. So this genetic drift or a random drift is also responsible for creating variability of croplands. We were discussing about the driving factors that was responsible for causing variability, and these driving factors, since it is causing resulting in variability, we can call those factors as evolutionary forces also, because that is responsible for evolution, crop evolution. So now coming to the sources of germplasm. When I say source of germplasm, we can, from these sources we can assemble complete germplasm. So that's these all the sources whatever I mentioned, around 5 sources, from these sources we can assemble the complete germplasm, crop germplasm. The prime source is the wild relatives. This wild relatives it will have a common ancestry with the croplands, and it is in the wild, but otherwise the most of the quality traits that is found in wild relatives are very essential for the use in plant breeding. And second important thing is the land races. When I say land races, this land races are very important component, and this primitive varieties of primitive cultivars, land races, are developed by the farmers for long years, and when I say long number of years, it is more than 100 years, 200 years, so many years were they are involved, and they went for selection, the farmers went for selection, and they have developed certain cultivars, and those cultivars, though it has not been used, that cultivars, commercially it has not been used, but those cultivars are called as land races. They are repository of a lot of positive traits. And next is obsolete cultivars. When I say obsolete, it means that these cultivars are not in use for the simple reason that in the plant breeding activity, these cultivars in the initial stage itself, it has been let off or it has not been used, obsolete. But still this obsolete cultivars, if you look at it, they are having lot of resources. And next important point is the breeding lines, advanced breeding lines, mutation and other products of plant breeding are also form an important source of germplasm. And last but not the least, that is modern high yielding cultivars also forms an important source of germplasm. And if you look at it, we need to sum it up, because the PGR, the plant genetic resources, has to be used sustainably. For that reason, a bit of we need to understand the past and the present and the future. When you look at the past, the green revolution, the very important activity that has resulted in increased a quantum jump of agriculture production, and that green revolution was caused because of the development of high yielding semi dwarf varieties. But these green revolution products, if you look at it, the high yielding varieties, the genetic bottleneck was that it had a very narrow genetic base, and because of that narrow genetic base, they were found to be susceptible to the biotic and abiotic stresses, and it was very difficult. It has succumbed, most of the high yielding varieties has succumbed to the stresses, and that has caused a lot of harm, that has caused a lot of stress to the agriculture production. And what we need to understand from that, what lesson we need to learn is that we need to look for the PGR, we need to look for a sustainable way forward by including the PGR. And I also stress the importance of land races, because these land races, which have been found widely in secondary center of origin, they are having a lot of potential, and those land races have genes for resistance to the frost. For example, in the case of potato, frost resistant, that was found in Wisconsin. So in many land races, if you see, they are having a lot of the genes for resistance, and those genes has to be used or tapped for developing new crop varieties. And next important point is yield maximization through the use of PGR. When I say yield maximization, we have achieved the yield to a maximum extent, but we need to understand that still, if we incorporate more of PGR, we'll collect maximum possible and get the best of the PGR, and we incorporate it into our plant breeding program, we can maximize the yield. And at what definitely we can maximize the yield, and even at the whatever the stress may be, we can maximize the yield, we can get the best possible from use of the PGR. And next important point is the biotic and abiotic stress beading. For the biotic and abiotic stress beading, the important raw material is the plant genetic resources. And last but very important thing is root architecture. Till now, the agricultural activity or the plant breeding activity focused on increasing the harvest index and ultimately increasing the agriculture production. It means that harvest index had been increased, but let us look at an important component, that is the below the soil, that is root architecture. If we look, or if we do a lot of research on root architecture, we can improve the agriculture production. The basic reason is that if the roots are capable to harvest maximum possible water, nutrients from the soil, definitely the crop productivity can be increased. And for this reason, we need to look for the plant genetic resources from the wide everywhere. We have to find out that the plant genetic resources, the best possible resources, we have to find out that where we can, what ways we can improve the root architecture. Thank you very much.